as Tom was saying, I used to work for a company called Metrion, but we've just spun out into a new ion channel CRO. So we're very excited to be here because it's the first time we've presented at SPS or even attended SPS. And one of the important parts of what we're trying to do, what we've seen coming down the path, of course, is CiPA. So much like Biosciences, we're a small company. We specialize in ion channel screening. We do the usual range of cell line developments, assay developments, but our focus is on gigaseal quality. So we do medium throughput, gigaseal quality, automated patch clamp, and manual patch clamp. And a significant part of that work that I'll talk about today is our CiPA panel. So we've been working on this for the last couple of years to get our panel together and to validate our assays and to help put our package and align it with what's coming from the CiPA initiative. So we've had some good introductions as to what CiPA is. Today you didn't say what it was not, Bernard, but what it is something good, we think, so a regulatory approach that has been embraced by the American agencies, European agencies, and the Japanese safety organizations. And the idea here is to get away from HERG, this complete reliance on HERG, because this has a big cost, as we've been pointed out to us today, that there are very promising drug discovery programs that have been halted. There's novel chemistry scaffolds that have been stopped because of the promiscuous HERG signal. And also some companies, some compounds and programs that got past HERG were then stopped because of a very sensitive signal in the QT assay. So I've always been thinking about this going forward, getting new compounds from new customers. But Bernard made an interesting point about being able to go back in time and try and rescue some very safe drugs that have adverse labeling on them. So that's a very interesting idea as well. So again, there's more cost benefit for the industry from this approach. So we know what CiPA is now. I hope you know what CiPA is now. So it's a three-part approach. I will be talking today about these two parts. So much like Daniel, Simon presented, you need to have a good panel of human cardiac ion channels, and preferably you want those running on a high-quality platform. And you will then be plugging that in vitro data, the IC50 data, into in silico models and making predictions about action potential prolongation and arrhythmia. And we will then be checking those predictions with data coming from human stem cell cardiomyocytes. I don't have time to talk about this, but I will be talking about these two aspects of our work. So what are the channels? Why have we chosen these 6 channels? For 4 reasons. A, they're all expressed in the human ventricle. For this reason, KB1.5 and HCM4 have been dropped from the panel. The second reason that there are known pharmacological targets for compounds that cause arrhythmia. The third reason is this graph here, because we have some compounds that we know block HERG, but they also block inward currents, such as the sodium and the calcium, and in this way they balance the risk, and there's no overall prolongation of the action potential or QT, therefore those drugs are safe. So this is the multiple ion channel effect. So we need to look at more than one channel, and preferably we need to look at all of the active channels in the human ventricle to balance the overall risk. That's why this is called a comprehensive initiative. The final reason is genetic, of course, that all of these channels here, the 6 CiPA channels, they are all associated with QT syndrome. So there's a genetic validation for the choice of those channels. And this is the ion channel working group. So these are the channels. So these are the inward currents and these are the outward currents. And we've heard some compounds block a little bit of this and a little bit of that, and in that way they're safe. Hopefully I'll remember to mention on a couple of later slides, there's actually the sodium channel is mentioned twice. So we're interested in both the peak and the persistent current. So this is the persistent current here. So we're beginning to think about how we can design assays for that, and there's also some thinking about how we can include that in the in silico models. And Gary Nerms has a poster, I forget what number it is, where he's talking about introducing a persistent sodium current in his action potential model. So those are the channels we need. It really shouldn't be a problem to study them now because we have these high throughput devices. This is an example very quickly about what the assays look like at Metrion. I'm presenting today on the assays that we've set up on the QPatch 48 system. Other gigaseal quality, medium throughput, automated patch platforms are available, but I'm just going to talk about the QPatch today. But this is very important that this is just talking about gigaseal quality. This is very important. This is not a qualitative effort. This is a quantitative effort. This is what CiPA requires, is real numbers to plug into in silico action potentials and to compare to plasma concentrations. It's very important. that we have good quality recordings that give us good quality data to make accurate predictions of cardiac risk. Why? Because we're doing away with a thorough QT. We cannot afford to be making any clinical mistakes here. So HERG, we've all seen these before. NAV 1.5, I'll go into a little bit of detail of that, we have some time. KER 2.1, the KV43, yeah, we had to hunt around for some good reference pharmacology for that. I think we used an Aventis compound, no offense Aventis, but KCNQ and HM4, we do have an assay for that, but it's fallen off of the CiPA list now. So the most important thing here is that we're not just concentrating on HERG anymore, we're concentrating on multiple ion channels. And the initial development of that was the multiple ion channel or mice effect. So this was originally published by Chantes, Jim Kramer in 2013, as Simon outlined. And this is an example of their validation of this technique. So if you look at the block of inward and outward currents in the core panel, so it's HERG, NAV, and CAV. If you just look at the block of a test set of about 30 or 40 compounds with known torsatogenic risk, if you just look at the HERG data, it's very hard to discriminate safe versus unsafe drugs if you're just looking at the HERG data. So this is their Model 1 from the Kramer publication. If you also now include CAV 1.2 data, then you do get a clear discrimination between safe, so these are non-torsinogenic and these are torsinogenic compounds. So this was a good validation for the approach, and obviously BSIS have embraced this and their customers have also embraced this, and now there's a lot more screening on this core panel. But there's another step now that we need for CiPA, and that is we have to go to the full panel of 6 ion channels. This is data from the ESI study. So this was presented at the QPatch meeting in England last year, and they've also published it this year. And they show, for example, just looking at the core channels here in red, but then also adding in KVLQT1 and KV4.3. This is a list of reference compounds. You'll see that quinidine, if you just use the core panel, as outlined by Chantest, is not identified as a risk compound. But once you add in the KVLQ2 data, then that is listed correctly as a risky compound. So we need to have this multiple ion channel approach to estimate the overall risk of compounds. And what's interesting here as well, it hasn't been mentioned yet, is not only is there this compensation or this balance between block of inward and outward currents, if you just rely on HERG, you're not looking at additional effects on other potassium channels which could increase your HERG effect. So if you have something that looks safe in the HERG assay but also blocks KVLQT1 or KV4.3, you're going to miss that. You're going to send that false negative through, and then you're going to have a very big and expensive surprise later on in your preclinical or your clinical development. So it's important to look at them all together. And the other one that's not listed here yet is the late sodium. So there's some examples of oncology compounds, antibodies that block kinases that seem to be blocking late sodium and producing arrhythmia, and also Chantest have got a poster upstairs where they're showing that their venlafaxine, so this is a mixed blocker, it blocks a lot of cardiac ion channels, but overall it doesn't produce arrhythmia, and they've got a poster upstairs showing that it also blocks the persistent sodium channel. So we need to include both the peak and the persistent. As I was saying, quality is important here. So originally, and I was there at the beginning, and I was screening HERG on the Ironworks in 2004 when I was at Exelixis. This sort of the confluence of the requirements for HERG screening and the low quality, high throughput devices have served the industry fairly well. We have not progressed any unsafe. We haven't sent any thorough QT actives through to the clinic. But it seems to me that what you need for CiPA is got to be better than that. You need to have good quality platforms. And this is my opinion. And I've picked some data that proves, I hope to you, that this is a valid approach. So this is data from Gary Murren's group in Oxford, so it's an in silico modeling group. And he was given unprecedented access to many years of Ironworks screening data, cardiac screening data from AstraZeneca and GSK in the UK. And he's showing for this particular compound, lahatamib, that it has a known action potential and QT effect of about 13 milliseconds and a known free drug concentration of a nanomolar. If you plug Ironworks data for the cardiac channels into his models, they do not predict that this is a risky compound. If you add in some Barracuda data, it probably gets a little bit worse. They will admit, in fact, at the QPatch user meeting a couple of weeks ago, that the guy from AZ said, yeah, the Barracuda is not that good. If you add a touch of manual patch, gigaseal quality data, then the in silico models do predict that this is a risky compound. So this is a good example of quality. You need gigaseal quality data, you need high quality data, and you're plugging in quantitative data into the in silico models, and then they're making predictions of QT risk and arrhythmia. So it's not like rank order, it's not like medicinal chemistry SAI. You need really good quality data. And this is why we're choosing to use the QPatch for our CiPA panel. The other issue that Simon touched on was choice of voltage protocols. I'll just concentrate here on nav 1.5. You're not actually supposed to see this in great detail, but the point being, this is from the CiPA slide. Only one of these reference compounds actually stimulate the sodium channel nav 1.5 at a cardiac rhythm, a sinus rhythm of 1 Hertz. There are people here that are using much higher frequencies and therefore getting higher potencies perhaps. So the choice of protocol is critical for all of the CiPA channels, but nav 1.5 is a good example of that. And this is what our assay looks like. So this is a 1 Hertz 10 pulse train, so we're using a sinus rhythm. We're just looking at resting block, but we're looking at a use-dependent mechanism here. So we can look at the block of the first peak as well as the block of the last peak. So we can look at phasic and tonic block, and we can get a use-dependent ratio. And so some compounds, in fact, a lot of compounds that block sodium channels have a use-dependent mechanism of action, and we can pick that up by seeing a shift in the IC50s. And this is not a very terribly sexy protocol, but it is a very physiological protocol. But even here, there are differences between the results that we get with this and what others have got, and I'll come to that a little bit later. So we've got our assays set up. We now also have a test set, 28 compounds that are recommended by the CiPA panels, a mixture of Redfern risks and compounds that Tim Kramer and Shan Test have identified, also Gary Miram's, and it's also based on adverse labeling from the FDA. So there's very low risk compounds, intermediate risk compounds, high risk compounds, the ones that circled here, the 6 that we chose to validate against our CiPA panel on the QPatch. We didn't do any more because we haven't yet got consensus on the voltage protocols we need to use for these cardiac assays. So we could do all 28, but we'd probably have to go and do them all over again once we select the voltage protocols. The good news, though, is that the HTS sub-team is meeting, and we've got 2 meetings in October where we hope to finalize the voltage protocols that we all use to validate our automated patch assays. So we ran these 6 compounds on the QPatch, and Eisai did a similar thing in 2014 and published this in 2015. So Metrion data is here, Eisai data is here, across the 6 channels. You can see here, as Simon was indicating, not a lot of activity against KER 2.1, a little bit of activity in someone with KB4.3, nothing against HCM4. The big difference was in the nav 1.5 potency. So these compounds are a lot less potent in our hands than they are in Eisai's hands. Very simple explanation for that. They stimulate 30 times more than we do at twice the frequency. So they're driving these compounds into a stronger use-dependent block. That's not physiological. So it would be good to think about physiological voltage protocols when we finalize the CiPA panel. But if you can do this many compounds in less than a week, and you can do this on a high throughput device, and you can get good quality data, this is really making the point that's in my title, that we can have cost-effective screening. So Bernard was saying, well, they're not going to screen every compound that the Pfizer chemists make. They're maybe going to choose to select this at the lead selection point. But our point is that as a CRO, we can offer very cost-effective screening. If you've got high success, high-quality assays, then you can put more compounds through there. And if you're taking the money out of the thorough QT, you can move it back upstream, higher up in your screening cascade to assess the risk of a wider variety of chemical scaffolding. Final thing is the in silico modeling. So we have our QPatch data. We can now plug it into the models. There's a number of different models available. This is from Gary Miram's slide. So he's comparing the Tentusha, which is a very simple algorithm, to the Grandi model, which is really based on rabbit ventricular, and the preferred model for CiPA is the O'Hara-Rudy. You'll see here that the Tentusha is very sensitive to KBLQT blockers, but not very sensitive to hERG blockers. The advantage of the O'Hara-Rudy is that it not only has prolongation with hERG blocker, but it also shows EADs, so ectopic action potentials after depolarizations. This is one of the main triggers for arrhythmia. And this is another example here showing the effects of desotalol. We know that it should look like this, that there should be a prolongation. No effect in the Tentusha model, and actually a shortening in the Grandi model. So this O'Hara-Rudy effectively recapitulates the known software from human ventricular tissue. So this is the preferred model, so that was the model that we used for our data. We accessed this model using Gary Miriam's recently published web portal, but as Bernard was saying, there could be an absolute profusion of in silico models. There's a lot of experts who say their model is better than somebody else's model. I'm very happy to hear that this will be standardized, that there will be one place that everybody can go to do this. Maybe there's an opportunity here for some cardiac expertise to help people interpret the data they put through this model, because not a lot of people are familiar with in silico modeling. So this was our experience, which was a very positive experience for us. It was very useful. So we put our test data from 6 compounds into a full model. This is very important that all 6 of the CiPA channels were included in the O'Hara-Rudy simulation by Gary Miriam's, and we can see a clear discrimination between the medium and high risk compounds versus the low risk compounds. So we see here the 2 type 1 stemazole cisapride, they produce prolongation, but they also produce very pronounced EADs. Petrogel is in a special class of its own. It's this sort of high risk, but not an EAD inducer, but it does produce prolongation. And if you just look at the data up to 10 micromolar, so if you go to very high concentrations, a lot of compounds will produce EADs. But if you have some sort of filter here, then clearly you can categorize high risk from low risk. So we have verapamil, the mixed blocker, pronalazine, another mixed blocker that also has some preference for the persistent sodium. They produce some prolongation but no EADs. So we get a qualitative discrimination between known high risk versus low risk compounds. And this is precisely the approach. And this is just a case study of what you would do with screening data and plug it into the CiPA and silico model and get predictions. And you would then test these predictions against data you get from human stem cell cardiomyocytes. So it seems very simple. It's been quite fun for us to do this over the last couple of months. So this is our qualitative summary that we can categorize compounds with known risk. It gets a little scary. We're going to use the same process to categorize compounds with an unknown risk, and we're then going to tell chemists and drug discovery companies that this is good, this is not good. The feeling from the industry seems to be we need a little bit more of a quantitative approach for this. We need to find a good, robust metric for predicting arrhythmia, and that's the whole point of CiPA. So we need something reliable. In the eSci paper, they looked at all the range of analysis that we have here. So this is just looking at hERG data here, and these are the low-risk compounds versus the high-risk in red. There's no discrimination if you just use hERG data. We all know that now. If you also look at the mice data, so they've used a similar analysis to CHAN-TEST, where this is a mixture of hERG and Cav 1.2, they don't see such a nice separation between low and high risk with their data set using their Q-patch data. And similarly, you don't see a good discrimination if you just look at changes in QT or action potential duration. But what eSci have is something very cool, which at the moment you do need a supercomputer, and we're not going to be able to run this from our desktop PCs, is they have a whole heart simulated ECG algorithm that run out of the supercomputer at the University of Tokyo, which is really, really cool. But I can't use it yet, which is a shame. But they see a clear discrimination here where they have simulated the ECG and measured, in a notional sense, the arrhythmia induced by 30 or 40 test compounds. And they see a very nice discrimination between high risk and low risk compounds. This is not the way. This is a very complicated approach. It's a very expensive approach. This is unfortunately not going to be the simple approach. So it would seem that we're going to rely on the arrhythmia predictions from the in silico models, and it would seem to me that really you want to have, perhaps use these EADs, not just a prolongation, but the actual ectopic after depolarizations. And I was happy to see that there's both Gary Miriam's posted upstairs that he's using, he's putting a threshold in here. What concentration does the compound start to produce ectopic action potentials actually after depolarizations? So again, he's selected this a parameter of arrhythmia potential to predict safe versus unsafe drugs. And I think that's definitely the way to go, and you can already see that we're getting data now that rationalizes and supports the approach that CiPA has been developing over the last 2 years. So this is very exciting for us. I was there in the beginning when we started doing hERG screening with high throughput devices, and that was 10 years ago. and it feels like we're on the verge of, as Bernard was talking about, the revolution. This is the second revolution in terms of in vitro cardiac screening, safety screening. But we've got a whole lot of extra tools here, which are very interesting in terms of a computational biophysical point of view, but also from a translational. We've now got access to these stem cell cardiomyocytes to test and hypothesis and refine our hypothesis, and I think that is really the very exciting thing. So I know Sophion are thinking about how they may implement recordings from stem cell cardiomyocytes. We're waiting for those breakthroughs, so no pressure, Tom. So that's it for today. That's just the summary, but I'd like to acknowledge Rob, who's in the room, who did all the QPAT screening, and also my colleague John Ridley, who did the in silico modeling with the web portal. We have poster number 31 if you'd like to come up tomorrow and ask some more questions. Thank you for your attention.